Hai, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Haris bin Maliki and I'm from entry 82204D. So in this video, I will present to you about a simple and rapid protocol for isolation of genomic DNA from oil palm leaf tissue, which is from this journal, which is I just only take from the DNA extraction. Okay, so for the introduction, we go. So the purpose for this method, which is it for increased yield and quality efforts for palm oil that have been started in order to meet the rising demand. And oil palm DNA can be extracted using a number of procedures that have been described for quickly preparing DNA from plant tissues. And although this technology has produced good quality and large amounts of DNA, so furthermore, it can also process a small number of sample at once. And the date palm extraction methodology described by Arif at 2010. And this was slightly modified in this investigation for routine isolation of appropriate quality and quantity of DNA from oil palm tissues. Okay, so for the method, which is I told you just now, which is the DNA extraction, we take the frozen Elaeis guineaceae, which is the oil palm tree leaves, using a modified method, which is just take from the frozen leaves. So the frozen leaves were ground, mixed with lysis buffer and centrifuge DNA was precipitated. Okay, so after that, we treated with the RNAs. So also the sodium acetate and cold isopropanol and resuspended in wash buffer. So the pellet was electrofrost in 0.8% agarose gels and visualized with 0.5 microgram ethidium bromide solution. Okay, so for the material, we can get about CTEC buffer, 2 gram sample of spare leaves, mortar, pastel, liquid nitrogen, sand. So as you can see from the slide, that's all for material we use to do the DNA extraction. And the results and discussion for this method, which is the DNA extraction of oil palm tree, the procedure seems convenient for routine DNA extraction from oil palm sample because it is quite straightforward and yields a substantial amount of DNA. And DNA quality and yield and were generally inconsistent, but when the same amount of the starting material, which is 0.1 gram, was treated using the unaltered RF technique, which is just now I mentioned. So additionally, the original methodology only used 0.1 gram of beginning tissues in 1.5 milliliter of Eppendorf tube with 500 pl of buffer. So which is the RF method was the modified one. So the conclusion we can get, this modified procedure offers larger tissue starting materials, higher genomic DNA yield, and simple, reliable processing, and it can process up to 12 samples per day. So, and yield similar quality to conventional methods. So, making it suitable for routine molecular biology, studies of oil palm and other plant species. So, we have a lot of method to do DNA extraction, but the ideal one, which is we can use the rapid method just by Arif 2010. So that's all I can conclude and give a feedback about this journal method. Thank you.